so I will call him back to the stage to present the T-cell class 1 tools binding, processing, and immunogenicity. Thanks, Jay.
Thanks. All right, so I'm gonna switch gears now, not too much, and talk about the T-cell class 1 tools, the binding, processing, and immunogenicity, with a focus on the next-gen tools. And we'll start with an overview of the endogenous antigen processing pathway for class 1, which we show in this little cartoon here, where we're trying to illustrate that proteins that are in a cell are just constantly being broken down by the proteasome. These proteins can be self-proteins or viral proteins or whatever proteins happen to be in the cell. They get broken down into smaller peptides. Some of those peptides can be loaded onto the TAP molecule for transport into the ER. Some of those peptides can bind to an MHC class 1 molecule and make their way to the cell surface. And some of those peptides can be recognized by a CD8 positive T cell to cause an immune response, which we refer to as immunogenicity. And so we kind of want to delineate the different parts of this pathway to show you what the different tools are predicting. The processing tool will predict this part of the pathway, the breakdown of the proteins into peptides and transport into the ER. MHC binding will predict binding of peptides to MHC class 1 molecules, and immunogenicity will predict recognition by a T cell. So we'll start with binding, which again is this part of the pathway. And for all of these tools, you can start from the same place. You just go to the T-cell class 1 link, where you can get to through either of those links. And we can start with a review of the class 1 MHC molecule. So class 1 MHC is expressed by most cells, and its function is to present peptides to CD8 positive T cells. It's composed of two chains. There's one invariant chain that's called B2M, or beta-2 microglobulin, and then there's an alpha chain that's a much longer chain and is extremely polymorphic with thousands of known alleles. If we look at the 3D structure, the binding groove is somewhat closed, and so this molecule can only accommodate peptides of a relatively fixed length between 8 and 11 amino acids. So for MHC binding predictions, there's a lot of data available, a lot of experimental data available in the IEDB that's been deposited over the years, much of it from Alex's lab, but also from many other labs, where the binding constants of peptides to various MHC molecules have been characterized in cost-intensive experiments, so cost-intensive in terms of time and money. And there's a treasure trove of data available that can be used to throw at machine learning algorithms to recapitulate the binding values. So to use the profiles of the peptides and their associated binding constants to be able to predict, to make predictions of the same. And so I will show you how to accomplish this using the T-cell class 1 tool on the NextGen Tools website. So you can start from the T-cell class 1 page, where you enter your antigen sequence into the input sequence box in one of multiple ways. So either typing it in, copying and pasting it in, dragging a text file. And when you do, you'll get a little bit of feedback down here that lets you know whether or not the format is recognized and how much data that you've entered. And after you've entered your sequence, the next thing to do is to specify the peptide lengths and alleles. And so you can do that with the peptide length slider and either the allele autocomplete or the allele finder. And as I alluded to in the previous presentation, one of the features of the allele finder is that it allows you to select our panel of 27 alleles, which you can learn more about at this link or in the paper that's linked there as well. And this is a set of 27 alleles that has been shown to cover about 97% of the world population. So if you have a peptide pool where one of your peptides binds at least one of each of these alleles, that peptide pool will cover about 97% of the human population. And to use that reference panel, I already showed this in the previous presentation, but you can click on the allele finder and hover over the allele panel button, click that and click submit. Oops. And then your allele, the allele area on the prediction page will be filled in with those alleles. And you can click away any that you don't want by just clicking on the little X's in those chips. In terms of length selection, the alleles, they differ in their preferences for lengths based on whether we're looking at binding or presentation. So what we're showing here on the left is the kind of preferred peptide lengths for 5 different alleles if we just consider binding. And we can see that the profiles, they look somewhat similar, but not too consistent all across the various alleles. Some peak at 9, some peak at 10. And so that's when we look at binding by itself. On the other side, if we look at peptides that have actually been eluded from the cell surface, so peptides that have gone through that whole pathway where they've been processed, they've bound to an MHC, they've made their way to the cell surface, and look at the same alleles, we can see there's a very sharp peak at 9, and all of the profiles look relatively similar. So I think that the take home here is that if you're going to limit yourself to one length when you do your predictions, you're definitely going to want to make sure that that is a length of 9. And that's why that's our default as well. So moving on with the example, we've entered our sequence, our peptide lengths, our alleles, and now we're going to go ahead and select a binding method. So we're selecting NMHC PAN 4.1BA, which is our recommended binding predictor. And we constantly or regularly evaluate the methods that we have available, and we make designations as to which methods are our recommended methods. And these will change as new data becomes available, and they have changed over the years. And these are the last several recommendations that we've had. So our current recommendation for a binding predictor is NMHC PAN 4.1BA. Our current recommendation for an EL or for a epitope predictor is NMHC PAN 4.1 EL. And we will continue to reevaluate and update our recommendations as benchmarks become available. And so Bjorn mentioned this yesterday, but we do have this automated benchmark system set up where as new data makes its way into the IEDB, new peptide binding data, we will take those data sets and run predictions with all of the methods that you see here and determine how well those methods can reconstruct the binding affinities or the binding scores for those peptides. And so based on that, our current recommendation is NMHC PAN 4.1BA, which as of last year was coming out on top in this benchmark. So we've now made all of our selections. We're going to go ahead and click run. When we do that, the input sequence gets broken up into all possible peptides of the chosen lengths. We also have an option called as is. There's a little checkbox there. I'm not sure if you caught it, but the purpose there is to allow you to input a list of peptides instead of a full-length sequence. And when you select that as is option, that list of peptides will not be altered. It won't be broken down. They will be sent to the tools at the lengths that as they were input into that box. So once we have our peptide lists, those get sent to the different tools and the binding affinity or elution scores are predicted. Then those affinities or elution scores are compared to a set of randomly selected peptides to assign a percentile rank, so to figure out where those scores or affinities lie within the distributions. And if you select multiple methods, you will get a median percentile rank returned as well. So when you hit run, you'll get this little loading logo, and eventually you'll get a table that looks like this. So I've removed some of the extra columns here just to kind of highlight the columns that come out of the binding affinity prediction. We have our animated CPAN BA IC50 and percentile rank. And by default, the output is ordered by the binding percentile, so the median binding percentile. And just to go into a little bit more detail on percentile ranks and their utility. So IC50 values, their distributions vary quite a bit from allele to allele. And so what I'm showing here is the predicted IC50 values for 10,000 randomly selected peptides for HLA-A0201. And we can see that most of the values lie towards the right of this distribution, where the good binders are kind of in this range over here. If we look at a different allele, HLA-A2402, and its predicted IC50 values, we see a similar shape to the distribution, but it's shifted a little bit over to the right. So if we want to figure out what is a good binder for each of those alleles, we can look at the percentile rank values to kind of normalize those two distributions. So I've drawn a line at percentile equals 1, and we're kind of zoom in on this area of the plot now. And we can see that at our recommended cutoff of percentile rank of 1, that HLA-A0201 has an IC50 value or a predicted IC50 value of about 27, whereas HLA-A2402 has a predicted IC50 value of 511. So by mapping the predicted IC50s to a percentile rank, it allows us to more easily compare binding affinities between alleles, and also, perhaps even more importantly, as Bjorn alluded to yesterday, is to be able to compare affinities between, or not compare affinities, but compare relative binding strengths between various methods. So we have our output, and I also showed this in the last presentation, but you can click on the display columns button to bring up this window and select or deselect columns and learn a little bit more about what each of the columns are. If you add more methods, you're going to get more columns, and this window now, or this control, becomes even more important because you can kind of remove some of the distracting columns from your display. I wanted to show this in the demo too, I forgot, but you can also download your prediction results. There's this little download button where you can download the output data in various formats, so the TSV, CSV, JSON. And you could download either the entire table or just the rows that have passed the filters that you set. And when you download either TSV or CSV, you'll get an output that looks like this, which you can open in Excel or your favorite spreadsheet editor. And I also showed this in the previous presentation, but some predictions might take a while. particularly with large inputs or a large number of parameters that you'd like to test. And so you can enter your name or a pipeline name and an email address and hit submit. And you will get an email with a link back to the pipeline, and you'll get an email when the pipeline completes with a result attached to it as well. So we've run our prediction, we have our output table. Now, how do we select the peptides that we want to move forward with? There's not one way that we recommend. There are multiple sane ways to do this, and we have a few recommendations for starting points. One is to pick peptides that are below a percentile rank of 1, where we know that that's going to capture the majority of binders for each allele. Instead of percentile rank, you can just set a percent cutoff. So if you predict 1,000 peptides and you want to take the top 1%, you take just the top 10 peptides for each allele and length combination. And instead of using either percentile rank or percent, you can use a affinity cutoff. So 500 nanomolar is kind of a classic threshold for binding affinity. You can also use an allele-specific binding affinity, which have been established over the years. And those allele-specific thresholds, you can find more about those through the documentation, which also links out to a Zendesk article with a table of the various alleles and their recommended allele affinity cutoffs, which were derived from this paper here. So our recommendations for this tool and for filtering, we don't have one recommended way to create your filters. There are a lot of reasonable approaches, and we've applied many various approaches over the years. You can combine thresholds, so a percent threshold or percentile threshold and an IC50 threshold. And also, in some past studies, we've established that you may want to use allele-specific thresholds as well. So that wraps up binding. Let's move on to processing, which is, again, this part of the pathway and starts from the same starting point. But now, instead of selecting a binding method, we've selected a processing method. And here, we're using what we call the basic processing method. On the legacy site, we had called this the combined predictor. And this tool will take into account MHC binding and a model for proteasomal cleavage and tap transport, and create an additive score to give you the likelihood that this peptide will be naturally processed. And if we zoom in on the various controls in this portion of the screen, there are a few options available for proteasomal cleavage, so which machinery to use. And by switching these options, it's going to just switch the motifs that are searched for in the flanking sequence around the peptide. And then there are options related to tap transport as well. And we leave those there for convenience, but our recommendation is to just leave those options at their defaults. So we've made all of our selections, and we're going to click Run. And we'll have an output table that looks a lot like the output for binding, except now we have columns that are related to processing. So we have a proteasome score and a tap score, and both of these, higher is better. And then we have an MHC score, which is just a transform of the binding percentile. So it's a negative log IC50 value. And the purpose there is, again, to make it so that higher is better, so that we can make these scores additive, which are the following columns. The processing score, which is just a sum of the proteasome and tap scores, and then the total score, which adds onto that the MHC score. So just a word on the performance and our recommendations here. Peptides that we know are high affinity binders to MHC molecules are also generally going to be efficiently processed due to the coevolution of MHC molecules with the proteasome and the TAP molecules. And so we recommend just using this tool kind of as an additional filter or to help understand why a peptide might not be a good ligand, even though there's strong binding. So this is kind of more of a companion tool for binding, which we recommend. As an alternative to using the processing tool, we recommend using tools that are trained on eluted ligand data. So the binding tools are trained on data that measure the affinity of a peptide to a specific MHC molecule, uh, all by itself, kind of in a vacuum. And whereas the MHC ligand dilution methods are trained on data where a peptide has already gone through that whole process of being processed, binding to an MHC, and making its way to the cell surface, and eventually being eluted in this assay. So those peptides contain signals from the entire pathway. And NetMHC PEN-EL is one of those methods, and that is what we recommend as our quote-unquote epitope predictor. We don't have this tool on the NextGen site at the moment, but we have developed this tool in the past couple of years called AXEL-F. And this is a tool that adds on to the binding predictions a bit by taking into account the relative expression of a peptide. And the idea is that a peptide, so what we're showing on the y-axis here are IC50 values, so good binders and poor binders, so from good binding to poor binding, and from low expression to high expression. And what we can see is that binders that are kind of moderate binders in this IC50 value range can kind of overcome that poor or moderate binding with a large dose. So if they are really highly expressed, we start to see a significant fraction of those peptides in the eluent. And so this tool takes both binding and expression into account. All right, moving on to immunogenicity prediction, which again is this part of the pathway. Again, same page, same links. So the immunogenicity predictor was developed based on a data set composed of both immunogenic and non-immunogenic peptides, and analyzing the frequency of amino acids within each of those sub-data sets. So within the immunogenic data set, it was observed that there's an enrichment of residues like tryptophan, phenylalanine, and isoleucine. And within the non-immunogenic data set, there's an enrichment of serines and methionines and lysines. And we can use those enrichments to analyze new data sets and calculate propensity scores. So what is the likelihood that a peptide will be immunogenic based on its amino acid composition? So just like the other tools, we are on the T-cell class 1 page, and now we've selected immunogenicity, and we get a option to or which positions we want to mask. So which positions do we not want to include in the evaluation? And generally, these should be the anchor residues of peptides, and we have the default set, the anchor residues for MHC peptide complexes, and we have these set at the defaults of the 1, 2, and C terminal peptides, which is the general rule, but we also have an option to either enter in the positions yourself as a comma-separated list, or there's an allele-specific option as well. So you can select whichever you please and hit run. And this should look really familiar by now. We have an output table that has one row per peptide and allele, and then the immunogenicity score, which again, higher is better, higher is more likely to be immunogenic. A word on usage of this tool. So although this score can discriminate between epitopes and non-epitopes, what we're showing on the right here is a cross-validation where we have the score for epitopes on the left and the score for non-epitopes on the right. And you can see the distributions are highly overlapping, although they're definitely shifted higher for epitopes than for non-epitopes. And in our cross-validation and on an independent blind set, we've achieved AUC values of 0.65 to 0.69. So it can discriminate, but those AUC scores are nowhere near the scores that we get with our binding predictors and EL predictors, which are over 0.95, 0.97. And so again, our recommendation here is to just use a cutoff of 0 if you're going to use this tool, and that will just add an additional filter to increase the specificity of your selected peptides. So throughout this presentation, I've shown you how to run each of the individual tools, but we can also run the tools simultaneously. So we can select, we can enter our sequence, make our peptide and allele selections, all 27 in the panel, and then start with our NetMHC-Pen 4.1 EL predictor, but now we can add on other predictors as well. So we can add, for instance, the binding, the processing predictor and immunogenicity predictor, and hit run. And after a little while, you'll end up with a very large table for this data set, 157,000 rows. Then you can whittle down with by setting various filters. So we can set the same filters that I showed in the previous presentation, a median binding percentile of 2%, and immunogenicity score greater than 0, and processing total score greater than 0. And we go from 157,000 rows down to 276 peptide allele combinations that we might want to follow up on. Now that you've run your predictions, you can then copy links and save these links to come back later or share them with collaborators. So either the full pipeline links with inputs and outputs or just the configuration links. So that's pretty much everything I wanted to show you. Just to review, our next-gen tool site hosts that complete T-cell class 1 suite. So we have the binding and elution, we have antigen processing, immunogenicity. We've established recommendations on our binding methods and guidelines on other methods, and those recommendations will continue to change as algorithms are evaluated, as new information becomes available. We don't have any uniform threshold that we recommend for selecting peptide candidates, but we do have some guidelines for some approaches we've used in the past. And with these tools, you can execute everything simultaneously and filter and share your results, and hope that it's useful to you. Thank you.